Thank you, Dr. Lin, for the very nice introduction. So cell-cell communication really plays important roles in many biological processes. So those communication may include, autocrine, in which a cell secretes some molecules, and these molecule binds to the receptor on the same cells, or could be paracrine and direct cell-cell interaction, or sometimes could be mediated by endocrines. So it has been widely reported those communication play important roles in cell differentiation, in tissue development, and in immune interaction between cells in disease. As one very famous example might be the immune checkpoint mediated by the PD-L1 and the PD-1. So those cell-cell communication could be computationally informed based on gene expression data, especially with the recent development of the single-cell RNA-seq technologies. We can infer the expression of the ligand genes in the sender cell and the expression of the receptor genes in the receiver cells. Then, based on computational modeling of the expression pattern of the ligands and receptors, we can infer the communication between individual pair of cells in a sample. So, however, by the time we start this project, our collaborator, Dr. Yuhua Chen, she really wants us to help analyze cell-cell communications mediated by metabolites. However, at that time, we found that there is no computational tool that can directly perform the analysis. So we decided to develop a new tool for this task. So the reason we want to develop this tool is we found in the community there has been lots of reports that those metabolites actually mediate lots of communications between cells in tissues. Meanwhile, our recent work also revealed the important role for some metabolite-mediated communications. For example, we found that the cholesterol actually mediate important signal for the hematopoietic stem cell differentiations. Well, our other collaborators, such as the lab of Dr. Chen, is also investigating those metabolite-mediated cell communications in disease, such as the atherosclerosis. So yeah, putting all this together, we have the background to do it. We have the appearances, and we know it's a very important question that need a tool in the communities. So we decided to develop this new algorithm, which we call the MEBOCOST. So as I said, by the time we started this project, there is no tool to detect the metabolite-mediated communications, largely because there is no database or curated list of metabolites that has been known to have a potential to mediate cell-cell communications. So there are very highly talented postdoc, Dr. Rongbin Zheng, in my lab. He started from initially developing computational algorithm to collect those reported metabolites that could mediate the communication, followed by manual curations. So I will not introduce too much detail into this process, but the result is he collected 160 highly reliable metabolites, for which both the enzyme, both the metabolic enzyme and the sensor protein for the metabolite has been reported. And those metabolite and the sensor genes constructed a set of 440 pairs of potential communications. So if we look at those metabolite and sensor partners that Rongbin has collected, you can say most of them are collected the first time from the literatures, while a few other has been annotated in some database, such as the Recon2 database, and some from the Wikipedia website and so on. But for all these partners, Rongbin and the other team members ensured that they have been referenced in reliable references. And we can also say the metabolite covered most of the major category of the metabolites. Well, for the sensor proteins, they can be divided into 3 major categories, including those receptor proteins on the cell surface, which means these receptors, they only bind the metabolite, but will not involve in metabolic reactions. There are also receptors in the nucleus and also cell surface transporters that only transport the metabolite into the cells. So then with the curated database of metabolites, their sensor proteins, and their metabolic enzymes, then we develop an algorithm which take the single-cell RNA-seq data as the input data, and then the algorithm extract the enzyme expression and the sensor expression. Then we perform the permutation to inform the likelihood of communication made by a pair of metabolite and sensors between single cells. And finally, Rongbin further added a flex balance analysis to further ensure that the metabolites is highly likely generated in the cells, in the sender cells involved in the communications. So then with that algorithm, the first thing we do is to perform some in silico simulation analysis to say whether MEBOCOST could really detect communications. So the first question we asked is it possible that this algorithm can always detect communication, no matter the data is real data or just some random noise data? So we added the random noise to real single-cell RNA-seq data. As you can see, when the noise to signal ratio is relatively low, the noise have little effect on the number of detected communications. However, when the noise to signal ratio increase twofold or higher, then the algorithm already detected no communications, suggesting that the communication we detected are from the real data rather than from the noise data. Also, Rongbin analyzed and found that the algorithm is relatively not that sensitive to the sequence depth, and also the performance is also linearly correlated with the sequence depth, such as the number of sequenced cells. So all these data suggest that the performance of the algorithm is very robust. So then next, Rongbin tested whether the communication detected by the algorithm is really biologically meaningful or functional. So we make use of a set of published colorectal cancer single-cell RNA-seq data sets, which also have a set of high-throughput critical screen data. So then when we run the algorithm on the single-cell RNA-seq data, we detected a set of metabolite-mediated communications between the cancer cells and the NK cells. Then from the high-throughput screen data, basically in that data, what the research have done is they knock out individual genes in the cancer cells and then co-culture the cancer cell with the NK cells to test the effects of the knockout of individual genes on the cancer cells before or after the co-culture with the NK cells. Because by default, the NK cells are supposed to kill the cancer cells. So what we say is when the genes involved in the metabolite-mediated communication between the cancer cell and the NK cells are knocked out, we found that the proliferate of the cancer cell will be significantly impaired, suggesting those communications mediated by the metabolites is likely protecting the cancer cells from being killed by the NK cell or suppressed by the NK cells. So to further test the effects of those communication, we also analyzed the TCGA colon cancer RNA-seq datasets. So what we found is the high expression of those genes involved in the communication between the cancer cell and the NK cells is associated with poor survival of the patients. So suggesting that those communication is protecting, also protecting the cancer cell from the immune systems, so that the cancer can grow better in the patient, causing a poor survival. So then we performed another test to say whether the cells that show communication mediated by metabolites are more likely to be close to each other in the tissue space. So the rationale is, as we can imagine, if the two cells are closer to each other, the metabolite secreted by one cell should be more likely to arrive at the other cells. So the cells that are closer to each other should be more likely to communicate with each other. So then Rongbin analyzed the multiple sets of single cell RNA-seq data sets. And he did observe, for example, in this example, you can say the x-axis indicates the distance between two cells. Basically, the larger value means the two cells are closer to each other. And the y-axis indicates the communication score between the cells. So you can say we observe a very strong correlation between the cell co-localization and the communication scores. So you can say overall, in the real data, the cells that show communication with each other are much more likely to be to show a correlation between their co-localization score and the communication scores. So then after our simulation and real data analysis show the robust performance of MEBOCOST, we applied to new data sets generated by our collaborator. But before that, we first run MEBOCOST on a set of public single cell data sets from adipocyte tissue in obesity patients. And we detected a widespread change of the communications between patients with low BMI and the patient with a high BMI. And interestingly, we found that macrophage appear to be a major cell types that show significant change of the communication between the obesity groups. So then we apply the algorithm to the brown adipocyte tissue single cell data sets generated by our collaborator, Dr. Yuhua Zhen's lab, and identified about 20 different cell types. So what's interesting is we found the number of communication mediated by metabolites seems likely to increase gradually in response to the cold treatment. And we found that the adipocytes seems likely to be the major cell types that are involved in those communication, no matter in communication sent out by adipocyte or received by adipocytes. So this figure further show, you can see the number of communication detected by the algorithm appear to be the largest in the adipocytes compared to the other cell types. So it's interesting because actually the number of adipocytes is not the largest in this data set because the protocol didn't capture all the adipocytes in these specific environments. For example, we know the vascular smooth muscle cell number and the endothelial cell number are much larger than the adipocyte cell number, but still we found the adipocytes mediate the largest number of communications, which is very interesting. And also we detect the change in many communications in response to the cold treatment of the mouse in the brown adipocyte tissues. One interesting example is this communication mediated by glutamate from other cell types to adipocyte. For example, here, the glutamate is produced by the endothelial cell and then received by adipocyte progenitor cell. And we found the communication increased gradually in response to the temperature, in response to the cold treatment of the mouse. So then Zongbin further analyzed those communication from other cell types to adipocytes, and he found the communication mediated by glutamate seems likely to be the strongest communications. So then we performed some experiments to verify. First, we verified whether the glutamate could really be produced by those cell types, because endothelial cell appear to be the one show the strongest communication. So we performed experiments in endothelial cell, and we verified it's true that the endothelial cell can produce lots of glutamate. Also, we treat the adipocyte progenitor cell with glutamate and found it can significantly promote the proliferation of the adipocyte cells. So next, we further tested whether the adipocyte produced by those cells can promote adipocyte differentiations. So the mark gene, the adipocyte mark gene expression level indicates that the differentiation of adipocyte was promoted by the treatment of glutamate. And also we found that the enzyme that produce glutamate was upregulated in the cold-treated brown adipocyte tissue from the mouse compared to the sub-nature condition. And but what's interesting is we found although the glutamate level was increased significantly in the brown adipocyte tissue upon cold treatments, actually the glutamate in the circulation system, which means in the blood, did not show a big change, suggesting that this effect is local effect in the brown adipocyte tissues. Also, we verified that when the glutamate enzyme was upregulated in the endothelial cells, the production of the glutamate will increase proportionally in the endothelial cells. So this data shows that the MEBOCOST could really reveal those biologically important metabolite-mediated cell communication that has been verified in our experiments. So next, we would like to introduce another very interesting gene, MECON, that we found to play important role in regulating the metabolite-mediated communications. So we are interested in this gene MECON because our recent work revealed that MECON is a very important endothelial cell gene that regulates both endothelial phenotypes and differentiations. So what we found interesting in the brown adipocyte tissue is among the endothelial cell in the brown adipocyte tissue, we found the endothelial cells show a highly heterogeneous expression level of the MECONs. So if you look at the expression of the endothelial marker gene CDH5, actually this gene, the marker gene expression appear to be highly uniform across the different endothelial cells. Well, for MECON, you can say there are three major groups of endothelial cells, including a group with high expression of MECON, a group with medium expression of MECON and a group with the lowest expression of MECONs. So this figure further show what's interesting is for the under-senior population that show the lowest expression of MECON, they start to express the dataset marker UCP1. Well, for the under-senior group that show the median expression level of MECON, they also expressed those smooth master cell marker genes. Well, for the under-senior cell with the highest expression of MECON, they didn't show expression for this dataset marker or smooth master cell markers. That's very interesting because Yuhua's lab has reported that a small subset of the smooth master cells actually could translate into the dataset cells. So this data seems likely indicate that MECON can protect the under-senior cell and prevent the transition towards the dataset lineage. So then we further perform the RNA velocity analysis to infer the potential transition between those cell identities. So interesting, we found for the under-senior cell with the lowest expression of MECON, this seems likely is transition towards the dataset progenitor cell populations. Well, for the under-senior cell with the median expression level of MECON, this seems likely show some transition towards the smooth master cell populations. Well, the under-senior cell with median or lowest expression of MECON seems likely comes from the under-senior cell with the highest expression of MECONs. So this data also seems likely indicates the under-senior cell, the MECON is protecting the under-senior cell identity and preventing the transition towards the dataset either directly or through the smooth master cell lineages. So next, we analyzed the change in the under-senior populations upon the cold treatment of the mouse in the brown dataset tissues. So interestingly, we found for the under-senior cell with a high expression of MECON, they didn't show big change of the population size or even show a little bit increase upon the cold treatment. However, for the under-senior cell with median or lowest expression of MECON, the population size decreased gradually in response to the cold treatment. So this figure is even more clear. So the middle group is a group with the highest expression of MECON. And this population was returned upon cold treatment, while the group with the lowest expression of MECON shows the most dramatic decrease of population size. Then we further analyzed the number of communications between under-senior cell and the adipocyte cell and other cells in the broad adipocyte tissues. So we can say for the under-senior cell with the highest expression of MECON, the number of communication between under-senior cell and the other cell actually is the smallest, while the population with the lowest expression of MECON show the greatest number of communication between under-senior cell and the other cells. So what's more interesting is when we knock down MECON in under-senior cells, we found those genes involved in the communication between under-senior cell and other cells was significantly upregulated, suggesting that MECON actually repress the genes involved in the communication between under-senior cell and other cells. So this figure further shows that no matter for the communication sent out from under-senior cell or received by under-senior cells, the number of communication is the greatest for the under-senior cell with the lowest expression of MECON, and the smallest in under-senior cell with the highest expression of MECON. So yeah, those results show that MECON is likely protecting under-senior identity by repressing the communication between under-senior cell and other cells. The kind of insulate under-senior cell from other cells in the tissue to protect the under-senior identity. So next, we perform lots of investigation about how MECON is really playing function in the under-senior cell lineage. So first we analyze the expression level of MECON and verified that the MECON expression level increased gradually during the endothelial differentiations or during the trans differentiation from fibroblast into the endothelial lineage. Also, when we analyze single cell RNA-seq data during the endothelial differentiations, we can say there is a population of a cell with high expression of MECONs. And we found this population is exactly the population that starts to express the endothelial marker genes such as KTR and the other genes such as CTH5 and so on, suggesting that MECON expression appear in the endothelial populations. And also this population size increased gradually during the endothelial differentiations. So then we performed the knockout of MECON in iPSC cells to perform differentiation experiments. So you can say that both the DNA sequence and the Western blot indicate the MECON expression has been knocked out. And as a result, we say that differentiation from iPSA to endothelial lineage was almost totally repressed. Before the lockout, we can get about 60% of endothelial cells after the differentiation. Well, after the lockout, we get only about 6%. Then we further directly knocked out the MECON in the endothelial cell. And I would like to say this is a partial knockout because it's a knockout of the gene in the endothelial cell, but we didn't pick a single clone. So it's a partial knockout similar to knockdowns. So then we investigated the phenotype, typical phenotype of endothelial cells. So for example, for the tubule formation, which is important for the generation of blood vessels or angiogenesis, you can say the partial knockout of MECON significantly impaired the tubule branching or the tubule length formed by endothelial cells. Also, the endothelial migration is a very important phenotype for angiogenesis or the generation of blood vessels. And we can say the migration of endothelial cell is significantly impaired by the knockout of MECONs. And also the typical metabolism function of endothelial cells, such as the uptake of LDL, and also the production of the nitrogens are both impaired by the knockout of the MECON genes. So this data all suggests that MECON is important to protect the endothelial identity and maintain the endothelial lineage. Then to explore the molecular mechanism by which MECON regulates endothelial differentiation or identity, we looked out the MECON expression in the endothelial, the primary endothelial HUVAC. And then we performed RNA-seq and detected thousands of upregulated genes or downregulated genes. So what we found is for those genes that are downregulated by the lockout of MECONs, they are highly enriched in those endothelial cell identity related pathways, such as the endothelial differentiation pathway here, and also the VEGF signaling pathway, which is the venous endothelial growth factor signaling pathways. So those pathways are directly associated with the endothelial differentiation because the VEGF is always used to induce the endothelial differentiations. Also, there is other pathways such as the angiogenesis pathway, which is the blood vessel development pathways. So they are also enriched in those genes downregulated by the lockdown of MECON, or actually, so those are the genes upregulated by MECON. In contrast, those genes that are upregulated by MECON lockout or repressed by MECON didn't show these enrichments. And also because MECON is a transcription factor, so we analyzed the ChIP-seq data for MECON. and found that those genes that show the binding of MECON in the promoter or enhancer are also highly enriched in those endothelial cell identity-related pathways, such as the VEGF signaling pathways, or the endothelial differentiation pathway, or blood vessel development pathway, and so on. So overall, the promoter that are bound by MECON or the enhancer that are bound by MECON both show high enrichment in the endothelial-related pathway, and also the genes whose expression was regulated by the MECON lockouts. So next, we further analyze the high-C data to check if the 3D genome organization can be regulated by MECON, because MECON is a transcription factors, and MECON bind both enhancer and promoters. So what we found is for those enhancers that show the binding of MECONs, they are much more likely to form the chromatin loop between enhancer and promoter in the endothelial cells compared to the stem cells. So this is a very good example. The NR2F2 is a very famous endothelial, the venous endothelial lineage factors. So you can say both the promoter of this gene and the upstream enhancers show the binding of MECON at these locations. So then when we analyze the history medication data, we can say the promoter marker H3K4 trimethylation are highly enriched in the promoter region bound by MECON, while the enhancer marker H3K27 acylation was highly enriched in the enhancer bound by MECONs. However, those modifications appear in the endothelial cell, but not in the embryonic stem cell. So in the embryonic stem cell, we only see the repressive modification H3K27 trimethylation in the promoters. Then if we look at the high-C data, it's very clear that there is a very strong loop in the endothelial cell between the enhancer and the promoter bound by MECON, while those loops was not observed in the embryonic stem cells. So this data suggesting that the binding of MECON on the enhancer and the promoter in the endothelial cell is associated with the formation of a chromatin loop between the enhancer and the promoters. So as a result, those genes, NR2F2 was upregulated in the endothelial cell compared to the embryonic stem cells. So then we further performed experiments to verify the importance of MECON in endothelial cells in animal models. So we used the zebrafish as the animal models to perform MECON lockouts. And as a result, we can say the endothelial-related phenotypes, including the angiogenesis and blood vessel phenotypes, are significantly impaired by the MECON lockouts, such as the ISV length and the sprouting of the ISVs in the zebrafish. So in summary, we developed a MEBOCOST to detect the metabolite-mediated communication between cells based on single-cell RNA-seq data sets, and in combination with the flex balance analysis. Then our simulation data, our CRISPR screen analysis, and spatial transcriptomics analysis all indicates that the communication detected by MEBOCOST is biologically important and functionally significant. For example, they protect the cancer cell from killing by the NK cells, and also is associated with patient survivals. Also, using the MEBOCOST algorithms, we found the metabolite-mediated communication changed substantially in thermogenesis in the brown adipocyte tissue of mice, and also in obesity patients during the development of obesity. And also those metabolite-mediated cell-cell communication analysis, along with our comprehensive experiments, revealed that glutamine is an important regulator of adipocytes differentiation and proliferations, and could be generated by other cell types in the brown adipocyte tissues. to promote adipocyte functions. Also, we revealed that the MECON tends to repress the metabolite-mediated communications between endothelial cell and other cell types, so as to protect the endothelial cell identities in the brown adipocyte tissues. And also our comprehensive experiments in cell level or in animal models both verified that MECON is a very important regulator of endothelial differentiation and functions. So finally, if I could have a couple of minutes, I would like to introduce a database or website developed by Zongbin. We call it Metabolite-Mediated cell-cell communication portals, in which Zongbin has applied the MEBOCOST to the human single-cell landscape, which include over 2 million single cells from over 200 cell types, from 13 tissue types and 56 disease states. And with this data, he detected over 200,000 communication events mediated by metabolites. So then he deposited those communication landscape into this web portal, so that any researchers, especially those researchers don't have the appetite to run software or write algorithms, so you can easily just visit this website. You can choose the cell type that you are interested in, and or search for the metabolites that you are interested in, or any sensor protein that you are interested in, or the cell type that serve as a receiver of the communications. Then the web portal will return the list of the communications and allow you to generate lots of figures with some like an event plot, network plot, dot plots, flow plot, and so on. So basically, just by visiting this website, you can generate almost publishable figures for the metabolites and the cell types that you are interested in. So finally, I would like to really appreciate the fantastic team members in my lab, especially Zongbin Chen, who conducted almost all the bioinformatics work related to the metabolite-mediated cell-cell communications. And also Dr. Jin Lu and Dr. Bo Xiao, who completed most of the bioinformatics work for the cell identity related research and the MECON related research. And also some lab members such as Dr. Yang Zhang and others in Dr. Yuhua Zhen's lab generated the metabolite, generated the brown adipocyte tissue single cell RNA-seq data and performed the wide bench experiments for the validation in this in the MEBOCOST project. And Dr. Shenju, Min Zhang, and other lab members in Dr. Nini Zhang's lab performed tons of wide bench experiments to verify the important function of MECON in the endothelial cell lineages. And also our other collaborators, such as the lab of Dr. Hong Chen, Dr. Longho Fang, and Dr. Jung Cook, performed lots of experiments in collaboration with us. And also we highly appreciate our founders. Okay, I will be more than happy to answer any questions. Thank you.
Okay, thank you, Dr. Chen, for the fantastic presentation. So now is our Q&A session. You are welcome to unmute yourself to ask question or use the chat function. And I will repeat the question to ask the speaker if you're using the chat function. Yeah. So there is one question from the chat. So do you see any potential expansion or update to the portal or MEBOCOST to further support metabolism research?
So do you mean the expansion towards other type of metabolic research, or still focus on metabolite-mediated cell communication?
Let me see in the chat. I guess he means like do you plan any update like in the future expansion for the tool? Like what's the
Oh, yeah.
future? Yeah.
Yes, yes, yes. We have plan for multiple expansions. For example, one thing is currently the web portal include only the human landscape. So definitely it's important to further generate the landscape for mouse single cell data and probably some other species. That's one thing we want to expand. The second thing is we are further increasing the number of curated metabolites that have a potential to mediate cell-cell communications. I think recently Zongbin has already doubled the number of the curated metabolites, and we will continue to further increase this. And also the web portal will continue to add new functions, such as the downstream genes that might be the downstream gene whose expression level might be regulated by the metabolites and so on. And there are much more we can do. Actually, Zongbin have many more fantastic ideas that he is currently pursuing. And another thing I really need to mention is the spatial angle of the metabolite-mediated cell-cell communications. So currently we are generating spatial transcriptomic data and also spatial metabolomics data. And Zongbin will further improve the algorithm to detect the spatial communication mediated by metabolites between those single cells.
Is that answer the question? Okay, yes, thank you. Any other more question from the audience? You want to unmute yourself or join the discussion? Please welcome. Okay, I see.
Hello?
Yes,
Hello?
go ahead.
Hi. Hi, Kaifu.
Hi, Jijun.
Hey, this is Hi. It's great talk. Thank you. Thank you for sharing. As I was working on other things, so I've missed a little bit of your first parts of MEBOCOST. So as I previously worked with Rongbin on when you're developing these tools, it was amazingly useful for our projects. So I would like to ask you, have Rongbin updated the MEBOCOST that now you can compare multiple groups in the single run, or still, I remember previously you need to run individual data sets first and then compare by manually by the profile of the receptor or ligand metabolite sensor coupling. So now can you compare them side by side in the single run?
Thank you. That's a great question, and also thank you for using the MEBOCOST algorithm before we publish it. Actually, your feedback has been very helpful for us to improve the algorithm. So regarding your question, from my recent discussion with Zongbin, I think he already added that function in his algorithm, and he showed that with that function to directly define the differential communication between samples. Regarding whether he has released the algorithms, I need to double check with him. If he already released, that's cool. You can try immediately.
Yeah.
Not yet. We can either try to release it as soon as possible, or we can share it with you to try it before we release it. Or Zongbin, I see Zongbin in the audience. Zongbin, if you are here, could you help answer the question?
Hi. Yeah.
Hi, Rongbin.
Yeah, Kaifu, you are right. I'm just I just added a function and generate the figure. I need to think about more ways to draw the figure so the researchers can better have a better idea to know what's really the differential. But we have done the algorithm to quantitatively measure the difference. So I think it's coming soon. Yeah, I hope we can do that in the coming month, so we can release very soon.
That's great. That's great. Yeah. Thank you, Rongbin.
Thank you for your question. Yeah.
Another question is, I'm wondering, because usually when we have like a single cell data sets, it's from an individual organ, right? It's from individual tissue or organs. So then when we want to study the cell-cell communication, we just using that data sets, and it's possibly the microenvironments we thought that the metabolites and the cell communicated in that organ. But it could also happen that the metabolites is coming from the blood, it's coming from other like cross-organ communications. So in that case, if I combine two organ data sets and then do the same prediction, and like one metabolite might synthesize in one organ and the receptor might express in another organ, then I guess the result can give us a lot of prediction. But how to you, how much confidence do you think that the algorithm can give us the information in this way?
Yeah, that's actually that's an important question, and that's a question multiple people has asked us before. So in theoretical, in theory, it's possible to detect those communication between different. So you are talking about communication between cells from different organs, right? Through the circulation system. So we think it's possible to detect them, but will be probably more difficult. That's because in the blood, in the circulation system, there are lots of metabolites there. And based on our experiments, sometimes the metabolites generated by the cell only have local effects. For example, the glutamate that we detected, it increased in the brown adipocyte tissue in response to temperature. But when we analyze the circulation system, we didn't see the increase of the metabolite. So I would say you could do the analysis using the algorithm, but need to be very careful and to validate them very carefully. I would say if you do the validation, the validated positive rate might reduce compared to those analyses based on cells within the tissues. And Zongbin actually did some experiments. He can do regression to infer the effect within tissue versus mediated by the circulation system and so on. So till now, what we are doing is we are trying to tell people, yes, there are some effects from the circulation system. But if you have metabolomics data from the circulation system, then we can use those data to regress out the effects from the blood system. But in future, maybe we can, improve the algorithm to more reliably detect communication between organs, given that user also have metabolomic data from the blood system. We believe if you have metabolomic data from the circulation system, the detection of communication between organs will be much more reliable.
Yeah. That makes a lot of sense. Thank you for sharing.
Question.
Yeah, thank you.
Yeah, thank you. Are there another question?
Yes.
Go ahead.
Hi, Kaifu. This is from Baylor. Yes.
So happy to see you here.
Yeah. So I have questions. Maybe I asked you before, but I maybe not. So we usually do circadian rhythms. We can isolate different cell type within, like in the liver. We can do metabolomics in one cell type, like a hepatocyte. We want to see another cell type, like a macrophage, respond to the another cell type's metabolites change. Can I direct, instead of input a single cell RNA-seq data, can I input just metabolomic data? Another is like back another cell type RNA-seq genes protein data. Will this work in your algorithm? Seems it's durable.
It's supposed to be durable. Actually, I discussed with Zongbin before because I was suggesting Zongbin to just treat circulation system as one cell type, so that can consider circulation system. The thing is currently the algorithm, we need to filter out the false positive events. Then to filter out the false positive events, we need to do permutation Then the permutation rely on multiple cell types to tell, okay, if I randomly shuffle the cell labels for the gene expression, how likely we will still see the detected events. So we use so basically that means we need multiple cell type as a background to tell the likelihood of false positive rate versus authentic events. So that's the only limitation. But if you don't worry about the false positive events, for example, if you are white bunch parameters, you know you will perform white bunch validation. Then I think, okay, you can still use it to calculate, to detect potential communication. Then you can further select some top candidates to do experiments to add add additional layer for screening. That's my suggestion. So Rongbin, how do you think?
Yeah, I think yeah, you're right. That's as what we discussed, that we, in order to perform a permutation test, we need a pool. I mean, we need a pool of cells to better estimate the background. So that's why that's a limitation. But in our manuscript, actually, one thing we applied, maybe useful to you, Dr. Guan, is that if you have multiple bulk-seq or bulk samples,
yeah,
so you can do a correlation analysis. In our analysis, when we identify some communication in single cell, we want to cross-validate what happened really in the tissue. We collect multiple bulk-seq sample in a synced system, because it's a bulk. Then you have multiple samples. Then theoretically you can do a correlation analysis between the metabolite enzyme and the sensor. In your case, is that if you have multiple metabolomics and multiple RNA-seq, so then you can calculate the correlation between the metabolite abundance versus the sensor gene expression. So I think that's also another way, but that also requires some multiple samples to have.
I think this is perfect because we do this kind of circadian rhythm. It's a kind of time course experiment. So we correlate the metabolite in one cell type, we correlate it with your sensor in another cell type. That's what we did for the ligand receptor analysis. I think that's kind of fit logical here. Okay, I can talk to you later. Maybe email you.
Sure.
Yeah, thank you.
Maybe if your project specifically need to analyze the bulk RNA-seq data, we can actually, Rongbin can help customize the algorithm to help.
Yeah, that's perfect.
Exactly.
Yeah, because we have this kind of time course correlation.
Yeah. So as long as you could have multiple samples, then the algorithm can still do the permutation based on the multiple samples.
Oh, that's cool. Yeah, thank you.
Okay, thank you. Are there any question and people want to ask question? There's one in the chat. Probably it's a simple one. So if someone wanted to start using MEBOCOST or the portal, are there any training material available to help them get started?
Yeah, for the MEBOCOST algorithm, Rongbin have the documentation, and we have also some helpful information in the website that we release, the GitHub website that we release the algorithm. For the web portal, Rongbin, do we already have something like a demo or documentation? I guess we are working on it, right?
Yeah, we are working on it. My plan is to make a video so that people can know how really we can play with the web tool.
The web portal, I think when I try the web portal website, it's very, very easy to use. It's just some like typical website. You just click, playing around it will be very easy to use it. But definitely we will try to provide some demo video to provide guidance.